Hi everyone, welcome to the tutorial on BBrowserX Pro, which is BBrowserX's platform for single-cell analysis and visualization. This tutorial will cover the essential steps of analyzing your own data, from uploading your data to cell type annotation to other downstream analyses. We'll focus on a specific case study combining two public studies and explain how you can gain biological insights from the analysis that you perform on BBrowserX Pro. Before we begin, I'd like to introduce the features that are available on the platform. For long-term and returning users, you'll recognize that we have another platform, BBrowserX. BBrowserX Pro is the upgrade to that with more advanced analytical tools and optimized processing times. One of the biggest motivators behind building a Pro version is the need to support very large data sets. And with the expansion of single-cell RNA sequencing, scientists are now producing atlases of data, which require more powerful processing and computing. With BBrowserX Pro, you're able to submit datasets of 10 million cells or more and visualize them on one UMAP, as you see here. Additionally, the platform provides a suite of basic visualization tools for you to examine the gene expression across different populations of cells. You can build violin charts, bar charts, dot plots, heat maps, and upset plots, to name a few. We also recognize that cell type annotation is one of the most rigorous and rate-limiting steps in single-cell analysis, and so we provided multiple ways for you to perform cell type annotation in an automated way that still allows for manual refinement. Some of the more basic analyses we provide include differential gene expression analysis, where you can find which genes are upregulated or downregulated in a group of cells. Following differential gene expression analysis, you can also perform pathway enrichment analysis, in which you select for multiple pathway databases to perform the enrichment on and visualize the results in a gene set enrichment analysis plot. Some of the more advanced analyses include pseudo-time or trajectory analysis, in which you can track gene expression changes across predicted cell states. This tool is particularly useful if you have naive and mature cell states, or if you have different disease states that progress in severity. We have a cell-cell communication tool which infers which cell types are likely to interact with each other just based off of the gene expression of their ligands and receptors. We can visualize ligand and receptor interactions and their associated pathways in different types of visualizations, like this heat map here. BBrowserX Pro not only supports the analysis of single-cell RNA sequencing data, but also perturb-seq data as well. When genes are perturbed or knocked out with CRISPR-Cas9, you can assess the efficiency of that guide RNA perturbation, as well as visualize enriched pathways of those perturbed genes. Finally, we have a cancer cell prediction tool, which predicts which cells are likely to be cancerous or malignant based off of their inferred copy number variation. When cells demonstrate a large upregulation or downregulation of proximal genes, these cells will have a high CNV score. When comparing this tool to spatially annotated data, we can see that cells on the right in blue are likely to be cancerous, and cells on the left in yellow have been spatially annotated to be cancerous. So we were able to validate the results with spatially annotated data. Therefore, this tool is quite useful in examining tumors, which are often composed of a mix of cancer cells and non-malignant stromal or immune cells. So that presents an overview of our features. Now let's dive into the demo. When you first open BBrowserX Pro, you land on the Workspace. This is where you can work in your personal workspace or in group workspaces with your other colleagues. You're able to work alongside your colleagues on the same dataset within these group workspaces and see each other's analyses. You can also organize your datasets by the species as well. And you notice that we organize data into projects and studies. Projects you can think of as a folder, and a study you can think of as a dataset. Therefore, you can house multiple studies within one project. If you don't yet have your own proprietary dataset, you can explore our public studies here. Additionally, not only would you be able to work in group workspaces, but you can share studies with individual colleagues as well. The Bookmarks tab is where you house all of the bookmarked studies from Talk2Data, which is BBrowserX's database of manually curated public datasets. Now I'll go over the 4 ways in which you can submit studies to BBrowserX Pro. The first way is for those of you who are already familiar with BBrowserX. You can simply migrate your studies from BBrowserX to BBrowserX Pro using the Migrate from BBrowserX button. You can either choose individual studies to migrate or select all of them to migrate them all at once. The next option for uploading data is quite simple. You can simply expand the file storage panel and upload your data directly from your local storage or desktop. The third way of submitting data is the way I'd recommend the most. If you have access to cloud storage, such as Amazon S3 or SFTP, you can directly mount your bucket to BBrowserX Pro and access your files that way. Lastly, for those of you with knowledge about programming at the command line, you can access instructions for BBrowserX Pro SDK here, where you can view the instructions for how to import the specific packages, explore your account, and upload and submit data. Now let's access the dataset that we'll be using in today's demo. We'll first create a new study, then we'll fill out the study details, the study name, the accession ID, which you can think of as a unique identifier created by you to help further organize your studies. We'll define the technology and create a new project to house the study in. I'll put the project name, and similarly, the project ID will help further organize your projects. You can upload one or more batches. We support a multitude of file types, as you can see here, and all you will need to do is select a file type and simply drag and drop it from the file storage into the field here. Our study includes 3 batches, one of which comes from a different dataset. Once we've finished uploading all of our batches, we'll move on to the QC and pre-processing. BBrowserX Pro offers 3 metrics to perform the initial quality control: the number of read counts, the number of gene counts, and the max percentage of mitochondrial genes. The platform pre-configures values for each metric, which reflect the standard quality control parameters. Once a study is successfully submitted, you may further change the parameters to be more stringent. Selecting the option to apply log normalization here will apply log normalization to your raw counts, allowing you to access both your raw and your normalized counts within the platform. For those of you who choose to submit an AND-data object or SURAT object, you may choose to skip the log normalization, and the platform will directly import the raw and normalized counts directly from the object. Once you've added the study, you can view the status of your study on the workspace. When the status changes to ready to use, we can now start exploring our study. To introduce the study first, these 3 datasets were taken from 2 sources: the Human Lung Cell Atlas from the Chan Zuckerberg Biohub SF with a PMID number, and the Haberman 2020 paper with a GSE number. You'll also notice that we have 2 PMID datasets. One was sequenced with 10x, and the other one with SmartSeq2. I plan to integrate all 3 and perform batch correction. These cells came from lung tissue, from both healthy subjects and those afflicted with pulmonary fibrosis. Pulmonary fibrosis is a disease characterized by fibrosis, or overgrowth of extracellular matrix in the lungs, where alveolar walls become thickened, thus impairing gas exchange. Some of the key players include epithelial cells and fibroblasts. In this demo, I'll be comparing the gene expression profiles between the 2 conditions by identifying potential targets and overall gaining a deeper understanding of disease mechanisms. Let's first go over some of the UI components on the Explorer. On the left-hand side, you have your suite of visualization tools and more downstream analysis tools such as differential expression and pseudo-time. At the top here, you have tools related to visualizing your single cells as a scatter plot and also annotating your cell types. On the left, we also have a gene query panel in which you can query for one or multiple genes and visualize them on your t-SNE or UMAP. Finally, on the right-hand side, you have your metadata panel in which you can flip through different metadata that you have. For example, the different conditions, samples, or cell types. I want to first talk about some of the things that occur in the background that apply to your study when you first submit it to the platform. This starts with feature selection and embedding. Because single-cell RNA sequencing data is high-dimensional and noisy, feature selection is a crucial step in identifying a subset of genes, otherwise known as features, that are most relevant for downstream analysis, thus producing more meaningful insights. A common feature selection approach is to select the top highly variable genes, and these are genes that vary significantly across cells because these genes are likely to contribute to differing cell states, conditions, etc. By default, BBrowserX Pro selects the top 2,000 highly variable genes. Next, embedding refers to the process of representing high-dimensional gene expression data in a lower dimensional space. This lower dimensional representation, or embedding, makes it easier to visualize, analyze, and interpret the complex patterns within the data. Typical embedding methods include principal component analysis, which aims to reduce the dimensionality of the data while capturing the most amount of variance in that data. That is, PCA will keep the most important features, otherwise known as principal components, of the data and reduce the noise. Batch correction can also be performed at this step. Since our study comes from different datasets, we'll perform batch correction across the datasets using Harmony, which is a fast general-purpose method for batch correction. Depending on the file type that you uploaded, the platform will either directly import the embedding from the file type or will automatically run embedding for you. Since we'll have to perform batch correction, let's run a new embedding. We'll click on Embedding and select Batch Correction as the embedding type. Then we'll select Harmony as the batch correction method and select Batch Name as the batch level metadata. Then click Run. Next, we'll choose to run a new t-SNE or UMAP based off of the embedding that we just performed. This allows us to visualize our single cells in a 2D scatter plot. Choosing the method of visualization depends on your study size. For example, t-SNEs are excellent at preserving the local structure of the data, but does not reliably preserve global structure. In other words, the distances between clusters may not be meaningful. This is more recommended for studies that are under or equal to 200,000 cells. For studies that are greater than 200,000 cells, we recommend visualizing your study as a UMAP. UMAPs excel at preserving local and global structures and provides a more accurate representation of the overall relationships between your single cells. Even though our study is only composed of 184,000 cells, I'll opt for visualizing my cells in a UMAP. I want to make sure I'm selecting an existing embedding, meaning I'll choose the embedding that we ran in the previous step. Now, when we compare our UMAP that is not batch corrected to our UMAP that is batch corrected, we can see that Harmony demonstrates a good integration of the three datasets. Keep in mind that, again, based off of the file type that you submit, the platform will either import the t-SNE or UMAP directly from your file, or will run a new t-SNE or UMAP based on the number of cells. Now let's move on to one of the most commonly asked questions when analyzing single-cell data. How do I know what cell types are present in my dataset? To tackle this, we'll start with clustering analysis. Clustering refers to grouping cells together based on how similar their gene expression profiles are. Typically, cells of the same type will have a similar expression profile, thus clustering will help identify distinct cell types. Louvain clustering is one of the most commonly used methods that constructs a network of communities or cell groups that both maximizes connections within a community and minimizes connections between communities. The resolution value determines the number of clusters produced. Lower resolution value will yield less clusters, where higher resolution values will yield more. The optimal resolution value depends on how finely you want to annotate your cells. If wanting to find cell subtypes or more rare cell types, a higher resolution value is more appropriate since clusters are more finely separated. I'll choose to input a resolution value of 0.5. Performing clustering at this resolution yielded a total of 24 clusters, and each cluster could represent a unique cell type. However, proximal clusters can also represent the same cell type. Now comes the task of cell type annotation. This is often a laborious task that relies on prior knowledge of key marker genes for each cell type. However, there doesn't exist a consensus list of key marker genes, nor can you find a list of marker genes for rare cell types. Thus, BBrowserX Pro offers an automated method for cell type annotation, only requiring manual review and refinement. The MetaReference method utilizes Talk2Data, our database of over 2,000 single-cell RNA sequencing studies, to compare the marker genes within the database against marker genes in your study. This method offers multiple metrics by which you can use to assess the quality of the predicted annotations, allowing you to manually refine the results based on these metrics and prior knowledge. We'll want to select the clustering analysis performed in the previous step as the input metadata. Then I'll choose to select subcell type as the prediction type. And this next step is very important. We want to customize the reference. This is because we don't want to use the entire database of single-cell RNA sequencing studies in our prediction, because this database is full of studies from many different tissues, many different cell types, and many different conditions. Therefore, I only want to include the most relevant studies in my cell type prediction. For this, I'll add lung tissue to the filter, then click Predict with Filter. Now let's go over how to interpret the results from the cell type prediction analysis. In the overview, you can see that for each cluster, it gives you a list of the predicted major cell types and the subcell types. Based off of all of the information and metrics that you gather from the results page, you can also change the final annotation. If we hover over one of these cell types, you'll be able to see a list of the core marker genes from our lung-related studies. Additionally, you can interpret the agreement rate as the percentage of studies that quote-unquote agree with this annotation of fibroblasts for cluster 1. Keep in mind that the percentage next to each predicted cell type reflects the agreement rate, and these percentages do not reflect the proportion of cells that annotate to these specific cell types. On the right-hand side, you'll be able to see for each cluster your list of the top 100 marker genes. Moving on to the Details tab, this is where you can get an overview of all of the studies that were used in the prediction. Hovering over one of the nodes, we can see how the authors have annotated this cluster within this particular dataset, as well as a list of the author's marker genes, the overlap score, the similarity score, and the rank of the selected gene within your cluster. If we were to click on a gene, this will navigate us to the Genes tab. This is where you can get more information about the statistics involved in calling this particular gene as a marker gene. For example, you'll be able to see the within cluster coverage, the outside cluster coverage, the log2 full change, as well as the weighted log2 full change. And finally, we have our Studies tab, which again gives you an overview of all of the studies that are involved in the prediction. If you click on the title, this will directly link you to the paper. After assessing all of these metrics and you're happy with the final annotation, you can choose to save these cell types to your metadata. And now when we navigate back, we're able to visualize all of these cell types on our UMAP. Some of the key insights we can gather from here is that the cell type prediction tool predicted a total of 17 unique cell types. Macrophages also appear to be the dominant cell type within this dataset. Now that we have our cell types annotated, we can visualize gene expression differences across our cell types, conditions, etc. This is where we can build basic visualizations for publication. If we click into the Basic Charts tool, then select the cell type metadata that was performed in the previous step as the x-axis metadata, and input Marco and Col1A1 into the y-axis gene panel, I can then visualize the expression of these two genes across my different cell types. Furthermore, I can add more information to each chart, not only visualize across cell types, but also across conditions. In the Split By tab, I can select my condition metadata. Here I can see that Marco, a receptor expressed mainly in alveolar macrophages, has slightly lower expression in pulmonary fibrosis when compared to normal. Our other gene, Col1A1, which is a collagen-producing gene, is most highly expressed in the fibroblast of lung cell types in pulmonary fibrosis, and this is indicative of overgrowth of extracellular matrix and fibrosis. In our visualization tool, we also have a cell proportion chart. This is quite useful in looking at how cell types are distributed across our different conditions. For example, here, when comparing pulmonary fibrosis versus normal, I see that capillary endothelial cells appear to be decreased in pulmonary fibrosis, indicative of lung injury and dysfunction. Another commonly asked question is, how do I identify the gene expression differences between my different conditions? This is where differential gene expression analysis can come into play. This analysis identifies genes that are upregulated or downregulated, in other words, differentially expressed in a population of cells. Highly differentially expressed genes often have biological significance as they could be drivers or contributors to a phenotype. Differential expression analysis is also ubiquitously used in marker gene analysis to find top marker genes distinguishing different cell types. For this, we offer many different methods to perform differential expression analysis. I'll opt to use the Pseudobulk DE-seq2 method, which is one of the standard methods in single-cell analysis that is often the most recommended because the results are more likely to reflect the biology that's occurring. This is because Pseudobulk will group together cells based on which sample they came from, thus we'll be able to perform the comparison at the sample level. In this analysis, I want to compare my macrophages in pulmonary fibrosis versus macrophages in normal. To choose my two groups, I can click on the composition tool. I'll generate my tree, choosing cell type as my level 1, and restricting the level to only display macrophages. And for level 2, I'll choose my conditions. Then I can assign my groups. In the results page, I can see my list of differentially expressed genes in a table on the left and a volcano plot on the right, which summarizes the gene table. Gas5 is among the most downregulated genes in pulmonary fibrosis, and it's been reported that Gas5 participation is recognized in asthma and lung cancer, but its role in pulmonary fibrosis is still unclear. Some mouse models suggest this long non-coding RNA attenuates PDGRF alpha and beta expression, which is important for the differentiation of pericytes to myofibroblasts, ultimately driving lung fibrosis. Overexpression of Gas5 inhibits this differentiation, identifying Gas5 as a potential therapeutic target. On the other hand, CCL2 is highly upregulated in pulmonary fibrosis and is known to play a critical role in the disease. CCL2 is the primary ligand for the CCR2 receptor and functions in mobilizing monocytes to sites of lung injury and inflammation. In mouse models, these recruited monocytes often differentiate into a macrophage subtype that adopt a pro-fibrotic phenotype. Following differential expression analysis, we can now perform pathway enrichment analysis, which takes our list of differentially expressed genes as input to identify biological pathways that are likely activated or suppressed in a population of cells. Our list of differentially expressed genes are first ranked based on a specific ranking metric called signal-to-noise. To understand the next steps that follow, first we'll run the pathway enrichment analysis. We can select all the pathway databases to be used in the enrichment analysis, and select signal-to-noise as the ranking metric. In the results page, we can see our list of enriched pathways on the left and what is called a gene set enrichment plot or a GC plot on the right. To interpret this plot, we can select up to 3 pathways to display. Each pathway is represented by a different color. Given a set of genes within a pathway, each gene is ordered based on where it overlaps with our ranked list of differentially expressed genes. You can see each pathway gene visualized by the bars here. For each pathway gene that is found within our ranked list of genes, the enrichment score will increase. The peak of the enrichment score graph represents the enrichment score value in the table. Finally, pathway gene sets that are ordered near the top or near the bottom of the ranked list are likely to be enriched. Thus, this analysis helps to understand the broader biological context and cellular behaviors that are occurring. BBrowserX Pro uses GC to perform the enrichment analysis. Some key insights from this plot are that interleukin-4 and interleukin-13 signaling and overview of pro-inflammatory and pro-fibrotic mediators from the Reactome and Wiki Pathways database, respectively, are positively enriched in pulmonary fibrosis, meaning they are likely upregulated or activated in pulmonary fibrosis. Next, the cell-cell communication tool infers cell interaction based on gene expression of particular ligands and receptors, and this is based off of the cell chat package. Sender cells with a high expression of the ligand gene and receiver cells with a high expression of the receptor genes will have a high interaction strength, meaning these two cell types are likely to interact with each other. We can perform cell-cell communication across our conditions and select the cell type metadata as the input. Here we can see that a lot of cell-cell communication is occurring between fibroblasts and type 1 pneumocytes in pulmonary fibrosis. This communication is likely occurring through this particular interaction from the collagen pathway, and this is most likely contributing to fibrosis and the overgrowth of the extracellular matrix. When compared to the cell-cell communication in pulmonary fibrosis, there appears to be less communication occurring with macrophages. If we were to hone in on some of these interactions, I'll be able to see the expression of these ligands and receptors across other cell types as well. In addition, I can get an overview of the interactions occurring across many different cell pairs, and these are most likely contributing to the overall cell-cell communication network. If we connect back to our differential expression analysis, we found that CCL2 was upregulated in pulmonary fibrosis. We can filter for interactions containing CCL2. While CCL2 has been found to be upregulated in pulmonary fibrosis macrophages, this gene expression is also high in fibroblasts, of both diseased and normal. Endothelial cells are the main receivers of CCL2 via the ACKR1 and ACKR2 receptors. These two are atypical receptors, meaning they do not function in initiating classical cell migration, such as in the case of CCL2 and CCR2. In fact, binding of CCL2 to ACKR1/2 may be competing for the binding to CCR2, thereby negatively impacting the overall pro-fibrotic and inflammatory response. Despite clustering cells together and annotating them as one cell type, one cell group still represents a heterogeneous population of cells at various stages of development, differentiation, or disease progression. Thus, it may be of interest to distinguish these various stages and identify key genes driving cellular transitions. Pseudo-time infers a trajectory along a population of cells to track the dynamic gene expression changes that occur across different cell states. This tool utilizes the Monocle 3 package. To start, we have to first subcluster the pulmonary fibrosis group. Next, we've identified that alveolar type 1 and type 2 cells exist within this dataset. Alveolar type 2 cells have been known to differentiate into alveolar type 1 cells in states of lung injury to restore the damaged alveolar lining. Thus, I'll further subcluster these two populations of cells to track this differentiation. I'll choose the UMAP corrected with harmony as the UMAP metadata. Next, I need to identify a root cell, or a starting point, located within the alveolar type 2 population. Genes involved in the cellular transition are found on the left. For example, SFTPC and SFTPA1 and 2 are standard and specific marker genes for AT2 cells. We see the decrease in these gene expressions as we arrive to our AT1 population. If we were to repeat the same steps in our normal condition, we can observe an overall lower proportion of both AT1 and AT2 cells in pulmonary fibrosis, indicating that AT2 cells are perhaps not able to differentiate into AT1 at a rate that could protect against fibrosis. Additionally, in our normal group, the AT2-AT1 population appears more difficult to distinguish from one another due to the proximity of these cell clusters and the gradual increased expression of SFTPC. Thus, the AT2 to AT1 differentiation may be occurring more in normal than in pulmonary fibrosis. As mentioned previously, the cancer cell prediction tool is mainly used to discern heterogeneous cell populations within a tumor. However, this tool can also be used to identify potential subtypes of a diseased population. For example, if a few subjects within a diseased population exhibit a proportion of cells predicted to be cancerous, this may be indicative of a more severe diseased phenotype, and it may be of interest to explore the subpopulation further. Before we can begin a new cancer cell prediction analysis, we'll need to combine two metadata categories together to see both the subject ID and the condition as one metadata category. I'll click the Manage Metadata button, add metadata, then select Combine Multi-Metadata. For Level 1, I'll choose Subject ID, and Level 2, Condition. Now I can see both metadata as one. Next, to perform a new cancer cell prediction analysis, we'll first have to define the cancer type. Since our study was taken from lung, we'll define the cancer type as carcinoma, which mainly affects epithelial cells. For the reference cell type, we'll let the tool choose automatically. This will choose a cell type that is assumed to be non-malignant, such as T cells, B cells, or smooth muscle cells, therefore setting a baseline for inferring the CNV of other cells. Under the advanced parameters, we want to make sure that we are selecting the non-batch corrected embedding, that is the PCA embedding. This is because we'll also be inputting the subject information, which will run cancer cell prediction separately on each subject. This is based off the principle that each subject has a different baseline or normal CNV. This also helps to identify CNVs that are unique to a particular subject. In the results page, you'll be able to see for each subject the inferred CNV scores. You can set the threshold for what you would consider a high CNV score, and the cancer cell labels will update accordingly. In the heat map tab, you can get an overview of the locations for where the CNV events are occurring and whether those locations are experiencing likely duplication or deletion events. In the phylogeny tab, you'll be able to see how groups of cells represented as a node branches into other groups of cells and gradually turn cancerous. You may click on a node to examine where on the UMAP that cluster of cells is located. If we were to look at the CNV scores across our subjects in a violin plot, we'll notice that subjects that were sequenced with SmartSeq2 appear to have a very high CNV score. This is most likely due to the increased sequencing depth and therefore are not relevant to compare to the rest of our subjects, which were sequenced with 10x. I observed that subjects 60 and 61 have a slightly higher CNV score than the rest of the pulmonary fibrosis subjects, which leads me to hypothesize whether these two subjects may be at a more severe disease state than others. In any case, it may be of interest to investigate further. We've now reached the end of our tutorial. We've covered the general workflow for analyzing single-cell RNA sequencing data on BBrowserX Pro, from data submission to cell type annotation, differential gene expression, and other advanced analyses. I hope this video has given you the foundation to navigate your own data analysis. And if you have any additional questions or need additional assistance, you can email us at support@bioturing.com. Thanks for following along.